we'll start. Okay, try to keep it on time. So this is our last seminar of the semester, and I'm very happy to have Ella Striem-Amit with us. Ella did her PhD with Amira Mehdi at Hadassah, Hebrew U, and then continued to do a postdoc at Harvard with Karmaza, right?
Mm-hmm.
And now for the past 2 years almost, right? She has been she's an assistant professor at the Georgetown Department of Neuroscience. And she has done really, really amazing work on plasticity and brain plasticity, showing how people, I really like the work with Amir, showing how people deal with a loss of vision, or and now she's working a lot of sensory motor systems and what happens with amputees. And I'm very happy to have you for the last meeting of the semester. So thanks, Ella, and the stage is yours.
Thank you. Thanks, Roy, for inviting me, and thank you all for joining me on this Zoomish seminar. Hope to one day soon meet you all in person. In the meantime, allow me to share my screen, if I can do that. And we'll dive right into the topic. As Roy said, I work on different aspects of brain plasticity. Today, I'll mostly be talking about the handless brain, the plasticity in people born without hands. So not amputees, people who have lost their hands later in life, but people born without them. And even though it's a Zoom seminar, please, 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 please jump in and ask questions. Unmute yourself and talk, otherwise I probably won't see you with all those tiny faces on the side. I would love for this to be more interactive. Okay, so let's begin. Generally, I'm talking today about hands. Now, hands are as handy as they can get, right? We use them to do everything in our everyday lives, to use tools to communicate with others. We need them to be integrated with our perceptual system, because we need to be able to grasp different objects and to understand others' actions. And we also need them to be coordinated so that we can execute actions. And we have multiple parts of our parietal and frontal and occipital cortices that are dedicated to the processing of things related to hands. And what I do is try to see what happens in the brains of people who don't have hands, who are born completely without hands, and who perform everyday actions to the same degree that we can, just using their feet. And to some degree better. I can't carve a pumpkin like she does. This is pretty darn impressive. Now, these kinds of human models, and I'll refer to them as dysplasic or dysmelics throughout my talks. I should be referring to them as individuals with dysplasia. I am trying to shift to that framing, and I consistently fail due to bad habits. So please don't take this personally, I'm trying. And these people allow us to test to what degree motor experience is crucial for hand-related brain functions. It allows us to test whether those neural systems that appear to process hands relate to the hand as a specific body part with specific visual features and motor features, and to what degree they're specific to the body part that performs actions, to the effector. In other words, to what degree is the organization limited by the sensory motor low-level features of hands? Now, to talk a bit about the individuals themselves, they're born without hands bilaterally with very little arm residues. I'm really, really selective in the individuals I pick for my studies. They have no hand phantoms. They are all right-footed, so they all perform daily actions with their feet. And this leaves me with an excruciatingly small sample. So if anyone here wants to talk about sample size and its limitations on studies, I'm very, very open to it. My approach generally says, if I am looking to see if you need hands in order to develop a brain structure, even one participant is enough. If I want to say that elephants can fly, one flying elephant should suffice for this. Now, I study a cohort of 5 individuals. I'm always recruiting, but that is a huge limitation of my studies. Overall, what we do in our lab is talk about and study congenital sensory and motor deprivation. So this means small samples in studying full and congenital deficits in people born blind, deaf, or without hands. And that really allows us to target those questions of dependence of sensory motor experience, the balance between nature and nurture, the extent and potential of plasticity, and the representational level at which those brain regions operate. Now, the work I'm presenting today was a collaboration between me and my fellows in Alfonso Caramatz's lab, who are each independent right now, Gilles, Moritz, and Alfonso himself, and of course some works done in my own lab, primarily by Yuchi, my postdoc who's moving to China to her own academic position, James, and Shri. I'll walk you through all those parts of the scene, beginning with visual motor representations. Now, we have a huge representation in our occipital-temporal cortex that's related to processing of body parts. And a large chunk of that, the part seen in yellow over here, I hope you can see my mouse. Okay, great, thank you, is dedicated to hand function, to processing seen hands. Now, importantly, this isn't just a visual area, because it is activated if we move our hands even without seeing them. So this is a visual motor representation of hands. And I wanted to see whether that would be found in people born without hands. And even more so, whether that area would show its typical association between hands and tools. There's a tiny region within that hand area that shows an overlap of preference between hands and tools. And that may sound trivial to you, except that body parts and objects, living and non-living visual objects, are separated in their neural substrates in the brain. So this overlap is non-trivial to some degree. Moreover, it seems to represent the degree to which tools extend the body. So this really has something to do with tool use. And we wanted to see whether that would be found in people born without hands who actually use their feet for tools every day. What we were able to see is that it is found, and you can see the individual peaks of every individual we studied here. For individuals with dysplasia in yellow and for control individuals in blue. It was found to begin with, and it was found in the same part of the brain to approximately the same size of overlap between the populations, with similar functional connectivity patterns to the rest of the brain, and similar pattern structure, representational similarity structure for that region. All of this suggests together that you don't need hands to develop this area. We were trying to come up with multiple, and reviewers helped us a great deal in coming up with alternative explanations of why this could still come out of motor simulation. So we know that this isn't about motor imagery because it's not activated when they move their feet. It just isn't. We know it doesn't have to do with phantom hands because they don't have any, or with prosthetic hands. So they haven't seen hand-like parts of their body manipulating tools because at least some of them did not use prosthetic hands at all. So overall, it looks like it develops it without experiencing or simulating hand tool use. Now, this tells us at least that the processing is not low-level motor. It's not attached to the hand low-level features. And that may not come as a great shock to you because we're talking about something that makes sense for it to be evolutionarily ingrained in our brains. Humans, or at least of the Homo family, have been using tools for generations. And I'm explaining this logic mostly for the benefit of the rest of my talk, because this is essentially the same kind of logic we apply to the rest of the data. We take this kind of data to show that brain organization can be evolutionarily conserved and doesn't depend as much on individual experience. We take this to be able to develop mostly through brain wiring and structure that develops it innately, prenatally, in fact, before the onset of motor experience, which means it doesn't really need experience to develop. Now, this doesn't mean that we believe motor experience has nothing to do with how we represent things in the brain or in our worlds, right? One of the core examples of where motor experience can come into play is in our representations of tools, right? And we can kind of try to think of what is it that makes an object into a tool for us. Part of it is definitely about its visual features. Tools are more elongated, they have affordances we can see and think of how we can manipulate. We know how they're used because we've seen other people use them, and we have experience in using them. Now, with individuals with dysplasia, we can rule out that last part and test what happens when they view objects that they can't use, have never used, as compared to tools that they have used. So really asking them on an individual level, with a huge list of 70 different objects, have you used this object? Have you used it with what body part? Even if you haven't used it, can you think of how you would use it? And coming up with, seriously, 3 individuals, because the other 2 could use every single item we have shown them, including the chainsaw. Scary concept there. And trying to see what areas of a brain distinguish between used and unusable objects in these individuals. And this really beautifully highlights the Proxy System, parieto-frontal regions in the brain that are engaged in action. We have the posterior STS, TPJ over here, frontal regions, even areas near the primary motor cortex that are able to decode in an individual with dysplasia which tools they can or cannot use. And the exact same regions show a difference between them and the typically developed individuals who can use these tools. I don't have this in the slide, but it also differentiates between the individuals with dysplasia who can use these tools and those who can't. So this isn't about handlessness. It isn't about whether you're using tools with your feet. It's just about whether you have motor experience in using that tool. Okay, so we've slightly walked through presentations of images of hands and tools. What is it that happens in action observation when we're viewing other people manipulate objects and perform actions? The reason we really wanted to target that is because of the well-regarded theory of mirror-neuron system, where it is thought, well, there are neurons who respond when monkeys and people view actions and when they perform them themselves. So we wanted to see what is the level of abstraction present in this mirror-neuron system. To what degree is that limited by the ability to simulate the seen actions? This is a study that actually targeted relatively high-level representations of actions. So we looked at the observation network and we were able to decode hand action type based on a very high level kind of representation. Is what the individual seeing opening or closing a trash bin or a door. So it's regardless of the visual features, it's regardless of the object that's being manipulated. Is it an open or closed action? And we could decode that in the ventral aspect, in the LOTC, in the area that we've just looked at before, and the interior parietal lobule in action observation nodes, suggesting that at least in these areas, at least at that level of representation, these areas are not limited by manipulation, by simulation of that sort. Now, together, we take this part of these research to show that at least parts of the association, ventral and dorsal visual cortex, develop the same visual functions regardless of what body part you're using as an effector, to act on the objects and to use for your own actions. And it's not limited by simulation, transcending those low-level motor features. Now, this would not come as a shock to you because your trivial response, and I'm almost surprised no one raised a hand to say that, well, these are visual regions, right? Why are you even looking at the motor functions of these regions? Well, personally for me, the reason I started looking into this is because in the years prior to studying these individuals, what I'd done was to show that some of these regions are not dependent on visual experience either, such that preference for body shapes, for tools, for hands, for actions, and for multiple other types of visual representations can be found in people born blind as well, suggesting that this isn't really about the features. It's not really about the visual features. And in our data, it's not really about the motor features. It really looks like the representation happening in those association areas is more metamodal. It's for the same type of computation. It's for a shape of a body part. It's for the hand. It's for tools. It's for reading. It's not for the visual features of script. It's not for the visual features of the body shape, transcending those features, and regardless of the visual or motor experience. So that's what brought me into this field. And I think that joining together the data from the individuals with the dysplasia and from the blind really allows us to say that the representation level in these regions is more abstract. It's not about the features. Now, this is a really nice model for perception. And we know that it also works for not only the association visual cortex, but also the association auditory cortex, because there are multiple studies showing that the deaf auditory cortex, at least the association auditory cortices, are organized for the same types of percepts, for language, even if it's through signing, for motion processing and localization, even if it's through vision and auto addition. But can this model also work for the motor system? How abstract is the representation of the motor system itself? And that brings me to the next question. Can this kind of metamodal model work for the motor system? Now, we know that in the frontal and in the parietal lobe, there are areas that are dedicated to different actions, specifically to reaching and to grasping. What we don't know well enough is what's these areas' core organization. Because these areas are engaged in reaching and grasping with our hands, and this is the body part we test to look for those actions, we don't really know what's the core organization. Is it an organization for body part? Is it a hand area that's as a byproduct or as a subspecialization organized for reaching? Or is it a reaching area that doesn't care what body part you're reaching with? In other words, if we have this model and the concept that the motor cortex may be organized for action types isn't new. It's been suggested by Michael Graziano and John Cass a while ago based on long trains of stimulation to the motor cortex, which seem to elicit complete motor actions and not just subparts of motor features. And using individuals with dysplasia, we can actually target and ask whether these areas are effector independent, whether they can function for the same types of actions, for reaching, for grasping, for manipulation, and so forth, if they're performed with the foot.
Can I ask you a question, Ella?
Of course. Thank you.
So I find this very interesting, and I was wondering, and it's you kind of hinted to it before, that when you say that these systems are abstract, that they respond, they're both kind of multimodal and they have an underlying computation, that some of this is not experience dependent either. So I'm wondering if you're saying that this reaching is something innate in the sense that it's really evolutionary. We have an evolutionary sense of reaching, and it doesn't depend. So if I wouldn't have feet and I wouldn't have hands or feet, I would still have a reaching area.
So definitely that's where I'm going towards. I have to say, this is not a claim I can make strongly, right? Because I don't have something that's essentially a brain in a jar. I've been testing people without input. I've been testing people with different outputs, but I don't have a brain that's completely devoid of experience. And I don't know what would happen to that. I don't know whether it depends in the dysplasics on visual experience and in the blind on motor experience. But I agree with you that the more parsimonious explanation would be that these areas develop innately to process to process and generate different types of evolutionarily guided actions. Now, we also have, and that's something that I won't show you today because it's still very much ongoing. We're also looking to what degree it depends on ethological origins, to what degree things like writing can have a motor action region, because we know that for reading, the visual word form area is completely found in the blind, but of course there it's also in blind Braille users. So it's a good question. 1 second. Sorry. Okay, so we're starting out by looking both at brain and at behavior, trying to see to what degree foot actions are even like hand actions. Does it make sense for them to have neural correlates and joint neural control? And looking at the neural substrates. So what we're doing right now in the lab, at least in days in which we can open, is have people grasp and reach with their feet and collect data about their foot movements and hand movements. This is an experiment we're doing with people in my lab and in collaboration with Erez Freud from York University. And we're really trying to see different aspects of the movement, the trajectory, the velocity profile, and the ability to pre-shape your foot to different object sizes. And I won't go too deeply into these findings, but generally we do find similar kinematics between the hand and the foot. And you can see the plots, the average plots are in thick lines and the individual plots are in thin lines. So you can really see that it's variable, but the hand and foot are very, very similar in their velocity profiles. Where we do find differences is in the deceleration, where it's less about the ballistic onset of the movement and more about the online control. And we're trying to see how that depends on effort and difficulty and experience. Now with COVID, it's been challenging to fly in the individuals with dysplasia, but we hope to do that as well. And in terms of pre-shaping, we know that we can pre-shape our hands even before reaching to the object, differently based on whether it's a large or a small object that we're grasping. And we can see the same happening for both the hand and the foot, where when you're grasping a larger bar, you're pre-shaping your foot to a larger degree, to a larger maximum grip aperture than for smaller objects. Again, there are differences. And obviously huge differences in scale, right? Because the hand can grasp more, it can, pre-shape to a larger degree, to a larger amplitude, and we're looking into that as well now. In terms of neural correlates, and we're also looking at individual differences in this, and this is really cool because I don't have the plot for that here, but it really looks like the individual patterns for velocity profiles and grasping for feet and hand are similar, such that if there's a pattern for control of reaching and grasping, it would be utilized within that individual similarly across the hand and the foot. So that's fun. Now, that all kind of brings us to say that it makes some sense for there to be joint control or planning in the brain for actions across the hand and the foot. And we were able to test that directly in having individuals with dysplasia grasp and reach with their foot, and individuals with controls, individual who are typically developed, grasp and reach with their hands. This is a really simple 2x2 ANOVA design, 2 actions, 2 groups. And we were looking separately at the effect of what body part and what group is performing the action and what action is being performed. So for the main effect of effector, hand versus foot and group, we find the typical suspects. We have sensory motor primary cortex for the foot. Also for the hand, it's just not seen in this map. But more importantly, for similarity in actions, areas that respond similarly for reaching with the hand and the foot than for grasping with the hand and the foot. We have areas in both the frontal cortex and the premotor dorsal, premotor ventral, and parietal lobe. as well as in the basal ganglia. And what's not seen here is the cerebellum. And even more fun than that, we have what looks like a gradient. So if the central sulcus is here, so these are the primary motor cortex primary motor cortex, where we expect to find main effective effector. So this is the foot area, the hand area would be around here. Closer to this region, we find preference for the body part. And as we go ahead into the frontal cortex to areas that could be more abstract, we do find the main effect of the action type across effectors. And we can really plot that as a graph between a posterior and anterior cortex, and seeing how that main effect of action rises more anteriorly, looking like a gradient of abstraction in the motor system. We have a parallel for that in the parietal lobe as well. And we're really digging into this kind of abstraction gradient in our ongoing current experiments now. Now, interestingly, we can also decode what action type is being performed in a lot of these areas in people born without hands. So these would be typically hand areas, and we can decode whether individuals born without hands are reaching or grasping with their foot. And we hope that, again, sometime along the line, we may be able to use decoding of action type to allow for use of brain-machine interfaces and prostheses. And the thought behind that is that currently a lot of prostheses are using fine motor control of timely grasping for objects with the right amount of force and so forth. If you go into the internet and try to look at demonstrations of people using hand prostheses, what you'll see is a hell of a lot of videos of people transporting eggs. Slowly and laboriously transporting eggs. And the reason for that is because that is probably the hardest task of doing it, of grasping at the right place in the right amount of force. And now the question is, to what degree we can replace that fine-tuned control with a larger, more abstract command of just grasp this egg and have visual algorithms compute the rest of the parameters needed, making it a lot simpler for prosthesis users. Now that's, again, way along these lines. I'm using this right now for 2 action types with fMRI, which is not the ideal tool, but it's a proof of concept. So I've walked you through perception and movement execution in the associations sensory motor cortex. What's going on in the primary sensory motor cortex? Now, Roy warned me that you also heard Avital's talk a while back. So this makes it more interactive, which is awesome. So as you may know from her talk, the 2 alternatives were either it's still topographic, right? It's a topographic sensory motor cortex. What we see in people with a late onset amputation and in late onset amputation models of animals is that it's overtaken by close by body parts. And that was not my preferred hypothesis, mind you. The alternative, of course, is that it would be reorganized to the body part used as a hand, suggesting that S1 and M1 are also as abstract and can plastically organize based on function. And that's what Tamar and Avital are working to show. Now, if I'll remind you, with my individuals, it gives us a very clear prediction of what body part would take over the hand area, right? I'll stop this before he begins singing. Great drummer, not that great in singing. So with these individuals, they use exactly 1 body part to replace their missing hands. So we have a very clear prediction. And we know that this area is activated when they move their feet. It's from both from Avital's paper, from a paper before that with an even smaller sample. This here is 5. So again, but my point is that it's reproducible. So they do move their feet and it activates their hand region. Unfortunately, what happened, and I was totally rooting against this hypothesis, is that in the hand area, which we localized with somatotopic mapping in controls, it's the area here in yellow, what we have is a total takeover by nearby body parts for simple movements. It's overtaken by the shoulder in green and by the abdomen in blue. And these are body parts that they really, really do not use to supplement for their missing hands. Now, I know that Kamar's group is working on multivariate pattern analysis to show that this area is relevant for compensatory use. I have some data that's really mixed in that respect. I think we really still don't have an answer to this question. But at least in this respect, the data looks like compensatory use and plasticity do not overcome the anatomical topographical constraints of this area. And the way I'd like to think of it isn't in terms of what body part it's being taken over, but in terms of that the computational level of this region is still tied to the sensory motor specific and is still not abstract enough. So as I showed you earlier for the premotor cortex, where we have a gradient of abstraction, I suspect that's what's going on. That somewhere along the motor system, we have areas that really are titrated to performing and to controlling specific body parts, which makes sense. At some point, we do need to dictate which body part is performing the action.
Sorry, could I ask another question about that?
Of course.
Okay, so I find that a bit confusing because if you think about the complexity of hand movements with their multiple degrees of freedom, so if you'd show me the lips and the tongue invading, that would make a lot more sense to me rather than the shoulder region kind of coming in, which has so few degrees of freedom and so different in the way we use it.
Yeah, I completely agree with you. Again, I did not foresee this result. So with relation to the face and lips, specifically, there's an anatomical border between the hand area and the face area that doesn't really have a lot of connectivity transcending. So I would expect that connectivity may limit the takeover in that respect. Whereas the rest of the organization for these regions is more consecutive. So, and one of the things we show with this data is that even in control individuals, there is some activation of the hand area for shoulder movement. So it's the dysplasic brain, much like the blind brain, is built on the same wiring principles as the typically developed brain. It just doesn't get the added experience later on, but it's still limited by the wiring that's prenatally there. And that's how we view it. Our brain is plastic to the degree that its wiring allows it. Okay, so we've looked at perception, we've looked at action. Overall, what we take all this data to mean is that these perceptual regions and computations develop without simulation, without motor experience. There are definitely some motor execution computations that develop without hands for the existing effectors, which looks like effector invariance. And overall, as a theoretical conclusion, it looks like the association cortices develop for the same functions. It's organized, abstracted from sensory motor features for the different perceptual computations that we need to perform and for the different manipulation and effector use and behaviors that we need to produce. We believe these are built on existing connectivity patterns. And we do acknowledge that there should be some limitations to where that computational selectivity model can apply to, in the sense that it looks like topographical area cortex are more limited in that respect. And as I told you, we hope to be able to, in the future, utilize some of those principles to augment prosthesis use. Now, in terms of where we're taking this now, as you asked about whether this is evolutionarily driven. So we're now looking at many more action types to try and parse out whether the entire action system is built this way or whether this is a reaching, grasping kind of thing, which can also be shown across the hands, for example, and across the hand and the eye, at least for reaching. And we're looking at writing as a model for non-ethological action and trying to see whether that's also develops as a mode of similar representation. We're looking more deeply into action observation, not only at the level of open and closed kind of distinction, but way more tuned and in a more active manner, having people view a hand action and imitate it with a foot and vice versa. And in terms of the abstraction gradient, we're looking at the cortical and subcortical loops, at parsing out action components, and at trying to make this a more clear model of how we can translate from a high-level action command to control of specific motor manipulation properties. Now, bear in mind that all this talk has so far been completely about handlessness. I also have a research line dedicated to reorganization and blindness and to sight restoration. And I'm actually giving a separate talk about this in the Bar-Ilan Vision Science Seminar in February 16, focusing on what's going on in primary visual cortex because of this, what are primary sensory areas doing in deprivation, and looking at individual differences there. Because in the blind, at least, I have a larger cohort to start probing into those questions of how consistent plasticity really is. If anyone's interested, we're actually looking for a postdoc with fMRI experience for these action abstraction kind of questions. So reach out to me if you're interested. And that's essentially it. I'd like to thank my collaborators, both in my postdoc and my current collaborators, my team, especially Yuchi, James, and Sri, and our participants who have been countless hours in the scanners and are just waiting for Corona to lift so they can fly and do that again. And thank you very much for your attention. Now, I would love questions. You guys have been so quiet.
Fantastic. So thanks a lot, Ella, first of all, for a very interesting talk. And yeah, let's open up to questions.
I would like to ask a question. You examined hand and legs, and you saw the similarity in the circuits. I wonder, if one lose his hand and they cut his hand, then actually some of the circuits can become more connected to the circuits of the legs somehow, and then actually the legs can take care of some of the functions. I mean, people that were not born without, they were born with. I mean, it will be interesting, to maybe to watch the process, how the connection forms, and this is very fascinating.
Thank you. I completely agree that this is very fascinating. And I completely agree with you that we'd expect a difference between early onset hand loss and congenital hand loss and later onset amputation. But I think if anything, what the literature has shown us, both in animal models and in works in amputees, and also in people who have lost their sight later on, is that the brain is less plastic in adulthood. And there can be less rewiring and less differences and less ability to learn to use those compensatory mechanisms. Now, I don't know if anyone has tried teaching amputees to use their feet, but I know that for all these individuals with dysplasia that I'm testing, they taught themselves. Their parents were to some degree on board, otherwise they couldn't have maintained those behaviors, but they really developed it on their own. Whereas people who have suffered an amputation later in life tend to use their either their remaining hands, their stump, their environment, and their mouth. And that's something that Tamar Makin's work has shown as well.
Yes, I predict that actually the circuits that are really related to the first motor layer of the sensing will be not changed. But I do think that there will be some more connectivity to higher level of decoding of the movement. So then, because this is in common to both, and then it will be able to. So I do think that there will be okay in the design of.
No, I agree with you. I agree with you, but I think we also see that in controls. We can also, what I haven't shown you here is the control data for effector independent grasping and reaching. And we get some of that as well, or at least at an interaction effect. So you're absolutely right that if we move our feet as if we were hand, we can use some hand motor representations to do that. The question is then, to what degree is it about motor imagery? Since I can't parse out these differences in what that would mean, I agree with you that it would be found, right? I just don't know what to make of it.
Yeah, I think you should find a patient that they just lost their hand, and then to use them for your research. It will be fascinating. Good luck.
Thank you.
Can I have a question, please?
Of course.
Maybe I missed it, but so you showed that the hand area is not the one supporting these superior feet abilities. So what is the neural substrate supporting these like super motor skills with the feet?
So are they super motor skills, or are they typical motor skills supported by an atypical body part? I don't, you're asking how I can localize the advantage, and I don't have really a control group for that. I have these individuals who use their feet. They use them as they typically do, and as we typically use our hands. Sorry, can you repeat that?
What task were they doing in the MRI?
Reaching and grasping. And now we're having them do more complicated tasks, like a bit of tool use and a bit of writing and all those kinds of things. So we can really look, but we will be looking at differences, right? So that could be a good way to go about that. We'll be looking at differences in fine-tuned motor parameters as well. So that may be an area where we can look at differences between individuals with dysplasia and controls. Yes, Michal?
Hi. Thanks for a really interesting talk. I wanted to ask or to discuss with you specifically the topic of gradients. But I have a little introduction to make about that. I think...
Shahli, please, I'm doing the talk. Please not right now, please. Sorry about that.
That's okay. So in fMRI, when we... Okay,
please close the door. I'm so sorry about this.
Well, I'm sorry. Okay. So in fMRI, when we see a gradient, maybe the earliest gradient that was demonstrated was a retinopathy, right? And in a retinopathy, you can find that in an individual. You move something along the field, and you can see the activation wave changing across the cortex. And then in more recent years, when people show gradients, in many cases, they skip this stage in which you show this in individual, and they primarily show this in a group image, in an analysis of many subjects together. And if you do that, it's quite possible that there is no gradient at the level of the individual, only at the level that you average these spots together across individuals. And because they're not exactly well aligned, you see something that looks like a blur in between two spots. So this was a very long introduction to asking, do you see these gradients in individual subjects, and are we clear that they're actually gradients rather than an effect of averaging subjects together?
Yes, so first of all, I completely agree with your point about gradients. that can form as a result of averaging. One of the advantages or disadvantages of working with such a small cohort is that we are constantly looking at consistency across individual subjects, and we're constantly looking at the individual subject maps. I can tell you that for the one place, so I can tell you that we have looked at that and it is the case in those individuals. But I also completely agree that it's important to continue looking at that at the individual subject level. One of the advantages of what we are hoping to do with parsing out movement aspects of like having grasp, but also having grasp and lift, also having grasp and lift and rotate, is to be able to parse out those differences and view all those maps at the individual level with a lot more detail. So I really hope to be able to support that in individual maps, or otherwise I really wouldn't believe it either.
Good. Thanks.
I have a question. Hi.
Hey, Bennett, how are you doing?
Hi. I have a question regarding the data on the topography gradient. You said that you didn't find differences in topographic mapping, but did you check in, for instance, connectivity differences? And more specifically, for instance, there are these data showing that there is strong connection between LOC and the hand area. So did you find this alteration for the foot area in the patient?
So you're asking specifically about the hand tool overlap area?
No, I'm asking because
generally, yeah.
Sorry, maybe I wasn't. My question is, okay, so maybe the topography doesn't change, but what can change is actually differences in higher level, like how the information is treated later on in later stages. And so one option is that, for instance, the foot area in this patient that actually used the foot as hands would show different connection, more similar to what in control we see for hand area, how it's connected to the rest of the cortices. So I'm asking if you're investigating, if you have investigated and what you found.
Yeah, I completely agree with you that, I mean, we come from the same theoretical background of ascribing brain organization and reorganization to wiring. So this is something that we're constantly looking at. I'm not sure I looked specifically at the functional connectivity from their foot area. We've looked at their connectivity from the hand area, from the hand tool overlap, and from these association motor cortices, which seems generally typical. There are some differences in how the visual cortex connects to the foot area, but not to the hand area in the motor cortex. So what we do see a bit of change in functional wiring, and we'd expect that. We do think that this is the underlying cause for the reorganization that we see.
Okay. Thanks.
Sure.
I have a question.
I think Dana is trying to raise her hand like a couple of times. So sorry, 1 second.
Thanks. So I was wondering if what would happen to the representation if a person tries to use a prosthesis, like to use something with the leg and then to use the prosthesis for the hand. Would the area, the representation area would be similar for both those that are used to use, let's say, their hands and will try to use the leg and vice versa?
I'm not sure I fully understand the question. You're talking about someone who was born without hands or someone who is a late onset amputee?
I refer to the two cases. So in the first case is the one that was born without hands. And what happens if this person tries to use the prosthesis at a later age? And is it similar for if any of us would be trying to use the legs to do those things, would the representation would be in the same area as well or not?
Great question. I can tell you that the individuals we studied really didn't like using prosthesis. Only a few of them were forced to try. Their parents forced them. They were forced to wear them just a few hours a week or so. They really didn't like it. We can't know if it's because of the specific prosthesis they were given, or because they were so much more useful. They were so much more capable with their feet. But they really don't use prosthesis, and it's a choice. They've tried, it doesn't work for them. We can't know whether it's because their brain has already so strongly wired the foot to those more associative representations, or what's going on over there. I'm assuming, again, my theoretical assumption is that we can really use these representations to act with any body part, or any body part that we want. How we can use it to use a tool like a prosthesis is a good question. That touches on kind of different aspects that are related to tool use and embodiment, and not exactly the same as using a different body part. As to controls using their own, individuals with a typical body scheme or typical development using their feet, we are doing that right now. We as a group, I can speak for myself, are not as good at using our feet for things, but we really can. There is a very small subsample of individuals who can't use their feet. They can't perform this action. Their toes move up and down instead of sideways, in a way. So that's a that's a low-level limitation to how we perform that action. But otherwise, we can grasp with our foot, with a big toe, and use things and write and do things pretty well. And that's without being amputated. Does that answer the question?
Yeah, so do you have any results for the second part?
Yeah, so
similar?
Yeah, it is similar. At least for grasping and reaching, we get interactions between the hand and action type that really works in the same areas as the main effect between the individuals with dysplasia and the control. So there really is such a gradient even in controls. These areas really are abstracted for action type, even for us. It's just that for us, we can't know whether it comes from motor imagery of the hand. And with the dysplasics, we can.
Yeah. Thanks.
Dee, you had a question?
Yeah, thanks. I wanted to ask, I understand that your participants are adults, but I was wondering if you would have access to an infant born without hands. Do you think that this reorganization or connectivity is developmental? Would its brain would be different? Would be similar to a typical brain? And then if you would be able to scan him for many years, would you see gradient changes?
So great question, and essentially my dream experiment. I would love to do that. Now, my assumption is, having viewed a couple of kids trying to grasp with their feet when they were tiny, we all kind of use our feet when we are learning to do fine motor control. If you put an infant on their back, which is what typically happens, and you put that università, that arch of activity stuff near their feet, which also typically happens because they move around, you'll see that they're trying to grasp things with their feet, and they're doing it, right? They're aiming their reach to the objects, they're trying to move them, they are using their feet to act. I would think that, if anything, the fact that we use our legs for other things later on makes us stop using our feet. In animal models, in primates, a lot of them do use their feet for different actions as well. So there's a lot of reason to expect that if an infant develops, even with hands, just in a way that allows them to continue exploring the world with their feet in the same way, they'd already developed some changes that develop the ability to use it and develop finer motor skills with the feet. And that would be extenuated, of course, in people born without hands, in infants born without hands. So my assumption is that this is all kind of an extreme version of the typical brain and its abilities. Does that answer the question?
Yes, thanks. And I was wondering, is there any data regarding animals that are born without limbs? Are they developing in a similar way to humans? And thinking about they don't have access to, I don't know, training or physiotherapy and stuff like that. How do they use the limbs?
Another great question. So I don't know of a study of an animal born completely without hands. There are studies of the early onset differentiation from John Cass's group, where they show plasticity and rewiring. They weren't looking at these association kind of regions in these guys. I've recently talked to John about this, and he said he'd be very curious as to really early onset loss of the hand. Again, I spoke to John, and he said that they don't amputate limbs of their animals because that would result in phantom limb pain. They only study animals whose limbs were amputated due to fights in the, the animal colony. So we can't really know. I would assume that the same would be found in these guys as well. You had a second part of your question about experience. And so there are interesting studies coming from Leah Krubitzer's group about environmental enrichment and its effect on plasticity and rewiring in blindness in animal models. I certainly expect experience and the ability to compensate using other body parts and using other sensory modalities in a full environment that really allows you to manipulate things with your foot, or to really allow you to perceive things through your other modalities, really affects brain organization. And that's what they find as well. But very much on target.
Thank you.
So, unless, so I have another question left. I was wondering what your view is kind of at the theoretical level. What is actually represented when you go forward, when you go apart? So you show this very nice gradient, and you could decode the action type independently of the effector in the more anterior portions, right? And this seems to go well with the data from other studies which actually do stimulation. I wonder what's actually represented in these premotor cortex. This is an action, is it a goal? What's your take on that?
So I don't know yet. Part of that is because I'm new to the motor domain. So I really don't know. I unfortunately do not yet know all the literature of these regions.
No one knows, that's okay.
I have that feeling too, but I can't say that with authority yet. I have a feeling that we still really don't know. There is evidence for gradients of hierarchical sequencing in these regions. So the complexity of the motor sequence that's being performed, as opposed to perhaps the goal, as opposed to perhaps more abstract representations of actions. I don't know exactly, and that's pretty much what we're doing right now, trying to parse those elements out.
Fair enough, fair enough. Cool. Any other questions? Okay. So Ella, thanks so much for coming. Let's give our speaker a big hand.
Thank you, guys.
It's a great talk. And yeah, so we're heading off to semester break. Everyone keep safe, keep working, and we'll meet everyone here at the second semester for a continuation of the seminars. Thank you all very much. Have a good vacation.
Thank you.
Bye-bye.